In this video, we're going to take a look at how different processes in meiosis lead to a wide range of variation in gametes. Basically, meiosis can create gametes that are very, very different from each other, and as a result of that, you can get offspring or kids that are very different from each other. And so you have a lot of diversity or variety in the population, which is very good for a healthy population. It's good that everyone's different, so that, let's say, for example, there's a disease that wipes one person out, it won't wipe everyone else out, since genetically there are differences that might make someone maybe more immune or have a better response or defense in the wake of that disease. We're also going to take a look at the process of gametogenesis, which is the process of creating gametes. As we saw, meiosis does that, but there's a bit of a difference between meiosis in males and in females, and we're going to take a look at what that difference is as well. So in these pictures you can see we have some offspring that are related to each other in that they're siblings. So here we have some brothers, and you'll notice that even though they're made from the same parents, same mom and same dad, that they look different from each other. So they're getting the same, they're getting genes from the same source, yet they look quite different. There are similarities, but they look quite different. And so, as mentioned before, meiosis creates variety of gametes, which leads to a variety of different individuals. And we're going to take a look at what leads to these differences in these siblings. Here you can see again some more siblings are related to each other and that they're brothers. Their DNA comes from the same source, same mom, same dad. There are similarities, but there are clearly some differences, and that's all because of the variety of gametes that meiosis can produce. And again, some more siblings over here. Same idea, different gametes, different combinations, differences in the siblings. They're not identical to each other. So here you can see me and my siblings. There are some similarities, but there are also some differences. And the main mechanisms that are responsible for the many types of gametes that exist are these two mechanisms over here. Independent assortment of homologous chromosomes during metaphase 1 of meiosis, crossing over during prophase 1, and this crossing over actually leads to a third. So the crossing over leads to a third, and we're going to call it independent alignment of chromatids and that's during metaphase 2. And so we'll take a look at what all this means, but essentially these are the three mechanisms that lead to the large diversity of gametes that are created by parents, which leads to the differences that you see in the siblings. That's why we're not more similar to each other. If it wasn't for these mechanisms, siblings would look a lot more similar to each other. So the first one we're going to talk about is called the law of independent assortment. Remember that homologous chromosomes pair up during meiosis. And so when they pair up, eventually they line up along the metaphase plate. And the idea I need you to get here is that when they line up at the metaphase plate, there's a few different possibilities of how they can line up. So you'll notice in this possibility, the blue lined up with the blue, and the red lined up with the red. And so if that possibility happens, then you get the gametes with the two blues in here, and the gametes with the two reds in here. Now, that's one possibility. The other possibility is that the big blue lined up with the small red, and the small red lined up with and the big red lined up with the little blue. And if that happens, you get gametes that look like this. You get big blue, little red, big red, little blue. And so you'll notice that you get 4 combinations, 4 possibilities of gametes because of the possibilities in the arrangement at the metaphase 1 plate. We call this the law of independent assortment because what it means is that the maternal and paternal chromosomes are independent of each other, meaning the blue ones, let's say the blue ones were all from dad, they don't have to line up with each other. The blue could line up with the red if it wanted to, which is what it did in this possibility here. So the homologous pairs don't care what the other homologous pairs are doing. This homologous pair will line up the way it wants, and this will line up the way it wants, which means it could line up with all blues on one side and reds on the other, or with blue and red and then red and blue. So they're independent of each other, and this is during metaphase 1. And as a result of that, it leads to a variety of combinations of gametes. If the blues always had to be with the blues on one side and the reds always with the reds on the other side, then you would only have 2 combinations in this case. You'd only have 2 combinations. But because the blues don't have to be with the blues and the reds don't have to be with the reds, you can have 2 possibilities. This can happen, or this can happen, which can lead to more combinations. The way to actually figure out how many possible combinations of gametes there are due to this law of independent assortment is using the formula 2 to the power of n, where n is the number of pairs of chromosomes. So for example, here we have 1, 2, 3, 4 chromosomes, which means 2 pairs. There's 2 pairs of chromosomes. So 2 to the power of 2 means 4 combinations or possibilities for the gametes that can be produced. And so this is the first mechanism that leads to a great deal of diversity in the gametes that can be produced. It's not just these 2 types here, it's now these 4 types that can be produced. You'll notice though that there is something missing in these pictures. If you look carefully, these cells here are from metaphase 2. And what you'll notice, and maybe you want to pause the video and try to figure out for yourself first, give you a hint, look at the chromatids in metaphase 2. Right? They shouldn't necessarily be identical to each other. Remember that there was crossing over that might have happened, so this blue one might have some red on it, right? This blue one here might have some red on it, and the reds might have some blue. So this picture doesn't even show the diversity that's caused by crossing over yet. That's what we'll go and take a look at next. That crossing over is the other mechanism that leads to the huge amount of diversity that can exist in the gametes. So this is just assuming no crossing over. But now with crossing over, there's even more different combinations of gametes that you can get, and therefore even more possibilities for your siblings looking different. So before we take a look at crossing over, let's just take a look at what are the possibilities for gametes with the following numbers. So if we have 2n is equal to 2, that means there are 2 chromosomes. That means 1 pair of chromosomes. So that means that there'd be 2 to the power of 1, which is 2 types of gametes. And you can draw it out to prove it to yourself. If you have 2n is equal to 4, that's 2 pairs. We saw it in the previous slide, but that would be 2 pairs. I'll just write 2p. So that would be 2 to the power of 2, which is 4 combinations. If you have 2n is equal to 6, that's 3 pairs. So that's 2 to the power of 3, which is 8. And hopefully you got this idea, is that the more pairs you have, the more different combinations that will be possible, the more different arrangements you can have at that metaphase 1 plate. For example, here I have 3 pairs, so I could have red, blue, red, blue, red, blue. So all blues on one side, all reds on one side. I could also maybe have 2 blues, 1 red, 2 reds, 1 blue. I could have it the other way around. I can have so many different combinations with more and more pairs. So this is us. This is 2n is equal to 46. That's 23 pairs. That means there's so many different ways to arrange the chromosomes to get those different gametes. So you can have actually 2 to the power of 23 different combinations of gametes. And again, this is assuming no crossing over. In reality, there is crossing over that happens, and sometimes many crossing over events that occur. So just without crossing over, you have 2 to the power of 23 different variety or different types of gametes that can be produced from just your, let's say, one parent, from, let's say, your father. And then there's 2 to the power of 23 different gametes that can be produced just from your mother. So now all these different combinations of gametes can lead to so many different combinations of genes in the kids, which is why your siblings, even though they look similar, will have so many diversities. In fact, it's almost weird that you look like your siblings at all with all these possibilities of gametes that can be produced. So let's take a look at the other mechanism, crossing over. Crossing over is basically just a reshuffling of genes during prophase 1. So let's say crossing over didn't happen. Here we have 2n is equal to 4. If 2n is equal to 4, that's 2 pairs of chromosomes, so that's 2 to the power of 2, which gives you, sorry, not 4 pairs, 2 pairs of chromosomes. So pair, which gives you 4 possibilities of arranging them. Now, if you take a look over here, this is our first possibility. This is our second possibility. Now, you also didn't see the other possibility at the metaphase plate. If you rearrange this to make all the blues lined up and all the reds lined up, you'd have 2 more possibilities, which would mean that you'd have 4 total. Now, if you take a look at this one over here, this is a picture on this side with 4 chromosomes again, so 2 to the power of 2. And that should give you 4 possibilities of gametes. Now, in reality, if you take a look carefully, there'll actually be 8 possibilities here. 8 possibilities. And the reason is because of this over here. Because of crossing over, these chromatids are no longer identical to each other. And since they're no longer identical to each other, the way they line up in the metaphase 2 plate will affect the different types that will affect the gamete produced. So for example, here we had the blue with little red and little red chromosome together. And here we had the red with a little bit of blue and the blue chromosome together. Now, these could have lined up in different ways. This blue could have lined up with that full red. So you could have also had a blue with a full little red gamete that could have been produced. So essentially, because the sister chromatids are no longer identical to each other, they can align in different ways along the metaphase 2 plate, and that leads to even more diversity. So without crossing over, you only have 4 possibilities for a scenario like this. But with crossing over with 4 chromosomes, you have 8 possibilities. And that's just assuming a little bit of crossing over happening. At least 8 possibilities that can occur. So crossing over just increases that amount of diversity. Imagine what that would do with crossing over events with 23 pairs of chromosomes. So you can see the idea here. Here we have the chromosomes lined up along the metaphase 2 plate. And in reality, these chromatids could align themselves in different ways. In mitosis, that can't really happen because there was no crossing over, so the sister chromatids are identical to each other. Whether you flip a chromosome around or not, it's still identical. But I'm going to redraw this cell over here, this one over here, with a different alignment of chromatids. So what I'll do is I'll put my long red one over here still, my short red one over here, but instead of the blue section being facing the top there, it's going to face the bottom here. And this one will still face this side over here. And so now what's going to happen is upon splitting, the big red, little blue ends up with the little red, and the little blue, little red ends up with the big red. Rather than big red, little red, and then the recombined ones here, you have big red, recombined, recombined, little red. So that's another set of gametes that can be produced, and the same thing can happen over here. And you get a different set. You can try drawing those out to see what you get if you just switch one of them around and see what type of gametes you get there. Usually the chromosomes that are made up of a combination due to crossover, they're called recombinant chromosomes because they had their DNA recombined. But this here we call it independent alignment of chromatids during metaphase 2. So because of crossing over, chromatids are no longer identical. Crossing over happened during prophase 1. Chromatids are no longer identical. So during metaphase 2, when they line up single file, the way they line up affects what gametes are produced. So that's why we call it independent alignment of chromatids during metaphase 2 of meiosis 2. And that's just another one of the mechanisms. It's related to crossing over, but you can kind of count it as a separate mechanism that leads to even more diversity in the gametes. The last source of diversity that exists that creates diversity in populations is essentially random fertilization. So we talked about how there's so many different types of sperm that can be produced and so many different types of eggs that can be produced. And because there's so many different types, there's so many different ways they can be combined. So I could have, let's say, sperm A combined with sperm B, or I could have sperm B combined with, sperm A combined with egg B. I could have sperm B combined with egg B. I could have sperm C combined with egg B, or there could be a different egg called egg C, and A could combine with C, and B can combine with C, and C can combine with C. Or I could have a sperm called sperm D because of so many different types of genes and recombinant DNA in there, and I could have an egg called egg A, and I can combine A and D, and A and C, and A and B. So there's so many different combinations of gametes because there's so many different types of gametes that exist. It's kind of like if you went to get an ice cream sundae. You want to build a sundae, and there's just so many different types you can build because there's so many different flavors of ice cream that exist. You can combine maybe chocolate, strawberry, and banana. You can combine maybe vanilla, banana, and strawberry, or vanilla, mint, and banana, or vanilla, chocolate, and strawberry. So you can think of these kind of like the different types of ice cream, and because there's so many different types of ice cream, there's so many different types of sundaes, your zygote that you can create as a result of that. So that's called random fertilization. Basically, any one of those many different types of gametes that are possible can be combined in so many different combinations, and as a result that leads to so much diversity even among siblings that are created from the same source of DNA, their two parents. So now you know why even siblings don't look alike. I mean, it's different when you're talking about twins. That's a whole different process, which you can research and add to your notes. But in terms of just regular siblings, here's why they don't look identical and why there's so many differences between them all. So this is a great animation that goes over the different types of the different mechanisms that lead to diversity. You can practice by doing the practice problem over here. You can also take a look at the animation, which will give you some tips on how to do this practice problem as well. So we're just going to finish off by taking a look at gametogenesis, which is the production of gametes in animals, and that happens by meiosis. But it's different in males and in females. So in males, gametogenesis has a special name. It's called spermatogenesis, basically the creation of sperm. You'll notice that here we have spermatogenesis on this side. You start off with a diploid cell, it goes through meiosis 1, meiosis 2, and you get 4 sperm with 23 chromosomes each that are haploid. And those 4 sperm can be used in reproduction and sexual reproduction. Now, this process is a little bit different for females. We're just going to briefly see what the difference is, the end result rather, and then in grade 12 you'll study more details on this process though. So the name for gametogenesis in females is called oogenesis, and it produces the ovum, or plural ova, so the eggs. And the eggs are also going to be haploid, but look at our difference here. During oogenesis, what happens is not all of the eggs survive. There's meiosis 1 that happens, and during meiosis 1, only one of the 2 cells produced will go on to produce other cells. The other one is going to be discarded. It's called a polar body. If the nutrients were split evenly, there wouldn't be enough in one egg to allow the organism, the zygote, to survive. So that's why one of the eggs just gets more of the nutrients and the components required. And then that egg goes on, divides, and that functional egg just produces one ovum, and the other one is a polar body. The first polar body also divides, but it's not used in reproduction. So the end result is that you do have 4 cells, it's just that 4 cells that are 3 of the 4 cells that are produced are called the polar bodies. They're not used, they're discarded, and only one of the cells produced is used in sexual reproduction. This is just for one cell, of course. There's more that are used, but the general idea is that you have one ovum, whereas in spermatogenesis you have 4 sperms produced that are fully functional. But in oogenesis, just one that's functional, and then 3 polar bodies, the end result. You want to make sure that you have enough nutrients to allow the zygote to grow and develop during fertilization. If you had them spread evenly, the nutrients, then it would not be enough to allow that organism to grow and survive.